Okay, so this is section 4. It's a small section, but it's super important, and it's really, really interesting. I'm going to say that about everything, so you'll have to bear with me. So, what determines the function of something is its structure, and why does it have to be structured a certain way? It's to perform a certain function, and we call that the principle of complementarity. And you're going to hear that if you take anatomy and physiology. You're going to hear that here in biochemistry. You hear that all over. Right? So structure determines function for DNA, and that's true. So if we look at a couple of different protein structures here, right? Let's look at these first. They have very similar structure, and they do also have very similar function. So if you look at them, they have the similar alpha helices, they have similar loops, they look very similar, and they perform similar functions. There are ribonucleotide reductase. So these are enzymes, and the one in red is found in bacteria, the one in green is found in mice. And so usually, if it has similar structure, it has similar function. And if you were to look at the DNA that codes for these two proteins, what you would likely see is that they're very, very highly conserved, which means they have the same base pairs in the same places. Let's look at a different example. Now we have a porin channel protein, the one on the left, the kind of short and fat one, and then we have green fluorescent protein on the right. So these two have very similar structures. We got beta sheets, we got loops, everything looks very similar, but they perform vastly different functions. The porin channel is to allow things to flow through it, and the green fluorescent protein is to fluoresce. So even though they have similar structures, does not mean that they're going to have exactly the same function, because a lot of times there are similar structures that can perform different functions. And it's just a small change that will allow you to do that. But what you're probably going to see is that the DNA of these two, I would say DNA is very similar. But here, even though our structure of the proteins is similar, I would say here that these two DNA sequences are going to be more different, right? And so you're going to end up with basically a very similar protein structure, but a very, very different function. So how do we get all these diverse protein structures? And really, it's evolution. It's nucleotide changes in the coding sequence of the DNA. And that's how we go from all these different proteins. So let's talk a little bit about that, how that works. If I can advance the slides. All right. So proteins are going to acquire all these different structures through random mutations of the DNA, right? We don't have mutations in the protein, we have mutations in the DNA. And I think I need to try and move this up. Oh, there we go. Well, close enough. Okay. So if you look at a wild type gene, wild type just means normal. This is unmutated. Normally, what would you see in the DNA? And we're given just a short stretch of that DNA, so just 3. And what we can see is that we translate it into alanine, glutamate, and serine. Now, if you look at this protein that's being made, in this case, it's an enzyme. And here we have a catalytic site. So this area right here, this is our catalytic site. And we have this side chain here sticking out into that catalytic site. So what you'll notice is that glutamate is negatively charged. Why would you want a negatively charged side chain of amino acids sticking out into a catalytic site? Well, what if what you're trying to bind is the opposite charge? So what if I have something coming in to bind, and that something is positively charged? Well, then that's going to help that attraction between the two, and we'll be able to latch on to that reactant, right, and catalyze the reaction. So that's great. Now, what happens if our DNA becomes mutated? Random mutation. And we have a single nucleotide change, and we change this guanine here to an adenine. Well, that's going to change your amino acid. So depending on where it is and what the nucleotide change is, sometimes it has an effect on the protein and sometimes it does not. So in this case, it does. It changes our glutamate to a lysine. That lysine has a very different functional side group. In this case, it has a positive charge. Well, if you have a positive charge and you're trying to attract something that has a positive charge, that's not going to work. You're not going to attract that substrate in order to have the reaction go. So this is going to end up being a defective enzyme. Not going to happen. So that's how we can have a mutation that's detrimental to a protein, to an organism, right? So these mutations can be beneficial and they can also be harmful. And under that, the mutations can be in what are called germline cells, and germline cells are egg and sperm. So anything that is passed on to the offspring. So the parent gets the mutation, passes it to the offspring. And this results in what we call inherited genetic diseases. Or it could be in our somatic cells. And so if you inherit a mutation in your somatic cells, like you go to a sun tanning booth, don't do that, don't do that. You go to a sun tanning booth and you end up with skin cancer, because that's what's going to happen. You're not going to give that to your offspring, right? That's in your skin cells. Those aren't germline cells. So that disease is now limited to the individual organism that acquired the mutation, okay? So how often does this occur? How often do mutations occur, and where do they occur? So if you look at a piece of DNA, and what you're going to see is the dark red portion. So this portion right here, this is the protein coding sequence. And usually flanking on either side of the protein coding sequence are regulatory sequences. So here's a regulatory sequence, here's a regulatory sequence. And then outside of that are non-coding pieces of DNA. And so if you look at the mutation rate in each of these different places, what you're going to see is our non-coding. So this is our gray. Our gray is the non-coding, non-regulatory. That area is going to accumulate mutations at a very rapid rate. But what you'll notice is that the regulatory and the coding sequences are not going to acquire mutations at a very rapid rate. So they're going to be very highly conserved. So if we take one particular protein, in this case, we're going to show you an example from G6P dehydrogenase. And so what we're looking at are different organisms. So we have bacteria, yeast, worm, fly, and human. And we compare all of the amino acids that are generated from this DNA. And so you can line them up. And so we see our first amino acid here, right, is the same for all of them. So all of them have a D, right? And so we say this is very highly conserved, and so we give it an asterisk. We say highly conserved, great. Now the next step down is, well, they're almost all the same, and any one that's different is very similar in structure in terms of the amino acid side chain. And so that's the two little dots. The next step from that is, let's see if we find another one right here, where we have only one dot. We say, okay, they're similar, but the one that's different, that side chain's very different. And then last but not least, you have something like right here where it varies a lot. And so we say that's not conserved. And so overall, if I look at all the sequences, there's a lot of conservation there. I see a lot of asterisks. I see a lot of conservation. And so what we would call these genes are orthologous genes. So in bacteria, yeast, worm, fly, human, they're all going to probably perform the same function. So they're orthologs. Okay. Now, how do we get genes that do different things? Because we have some genes that are different than yeast, right? But we're actually really super similar to yeast, FYI. But how are we going to get variations? Well, one way that happens is through gene duplication. So you start with one gene in an original organism. We call that the ancestral gene. And when the DNA is replicated, accidentally, that gene gets duplicated. And it happens. So one of three things happens once that duplication occurs. Sometimes if you have 2 copies of the gene, you can make twice the amount of protein, and that is yay, wonderful. And if 2 genes are better than 1, you keep them both, and that organism that duplicated that gene has a higher fitness, higher survival rate, and so it stays in the gene pool. Sometimes you only need 1. We only need this one. It's not really beneficial to have a second. And so that second gene acquires mutations. And because of those mutations, it gains a new function. And now, because it's gained that new function, it's given the organism a benefit. It said, oh, now I can make this new different protein that does something that the other ones couldn't, right? And so now that organism survives. That's great. The third option is that only 1 gene is required, and the gene does not acquire mutations that changes its function and therefore is lost. And so you go back to the original of only having 1. So you look just like the ancestral again. So those are 3 different things that can happen. So when we talk about genes and how they're conserved, how similar they are from one to another, anytime we talk about that, whether we're saying these are 2 genes in humans that were duplicated, right? And we say, okay, they were duplicated, so here's the human GR, here's human PR, right? Okay, well, these both came from an ancestral gene, 1 ancestral gene, but it was duplicated. So these 2 are in the same organism, and they're homologous, they're homologs. But there's a particular extra name that we put on it when they're in the same organism. When they're in the same organism, 1 species, not organism, but 1 species, we call it a paralog. Now, if we compare human to, let's say, mosquito, and we say, well, how similar is a human gene versus a mosquito gene? Well, then we say, okay, they're orthologs. They're very similar, but they're in different organisms. That's orthologs. So orthologs are different species, but genes are similar. Okay. So no matter if we're talking about a paralog or an ortholog, we're always talking about homologous genes. Paralog are homologous genes within 1 species. Orthologs are homologous genes within 2 different species. And that is the end of 4.1 and the end of the chapter. Yay! 1 down. See y'all next time. Maybe. Okay.